Thank you very much. Thank you, everybody. It's a pleasure to be here and speak before you all. This talk is entitled Single-Cell Nanophotonic Cavity Probes, and I'm Gary Shambat. My former advisor was Jelena Vukovic, and I led this work down at Stanford recently. So I'll begin with a brief introduction. Here is a picture of the human body. It's a fascinating biological machine. If we zoom in closer, our body consists of many organs. These organs are built of many tissue types, and within these tissue types we have many cells. And this fundamental unit, we have tens of trillions or so of cells in our body, and these cells determine all of our biological functions, ranging from disease progression, drug response, and cancer. So recently, many people have been interested in probing and understanding these single cells at the nanoscale. And on this slide alone, I show several types of nanoprobes. At Stanford, on the left side here, the Choi group has demonstrated electrical nanopillars for probing heart cells at the single cell level for measuring action potentials. The Milosh group has demonstrated nanostraws for single cell cargo delivery or fluidic access. And across the bay in Berkeley, the Yang group has demonstrated nanowires attached to fiber tapers for sub-diffraction limited spot size imaging of light and collection of light within an individual cell. Now, we took a look at this probe here and said, well, this is nice, but it's a relatively simple optical probe. Why not insert something more sophisticated, a more sophisticated engineered photonic structure? Why not insert a photonic crystal cavity inside a cell? Surely there must be some interesting applications. And so that is what we want to do. I want to take a photonic crystal cavity and insert it inside a single biological cell. Namely, I want to insert a nanobeam photonic crystal cavity, which has this needle-like aspect ratio for penetrating a single cell without too much damage. Now, what could we do? Well, we could functionalize that surface and do something like perform label-free sensing of proteins or DNA molecules. Now, normally when we do sensing experiments, we bring the biology to the sensor or the biology to the experiment. We take a sample or a fluid and we wash it over a bulky and mobile sensor. I want to do something very different here. I want to bring that sensor to the biology, namely to the single cell level. So what could we do? Let's say we have drug A, which is hypothesized to stop the progression of disease B by turning on the production of protein C. Well, if we have this probe inside the cell monitoring for this protein at the whole time, we can have a very interesting modality for measuring live dynamics of protein expression. So that was the basis behind this recent work published in Nanoletters last year. And this is what we do. We spent a lot of time over the last few years optimizing methods to attach semiconductor templates to the tips of optical fibers. Now, I won't go into the nitty-gritty details here, but show a simplified process. Basically, we use semiconductor manufacturing to fabricate these templates on the top surfaces of gallium arsenide chips. And then we take a small bit of general-purpose epoxy, attach it to the edge of an optical fiber, and essentially pluck out these large templates so that we have a final product, which is this spear or bayonet type structure with an optical cavity at the tip of this structure. I like to call these devices cellPC probes because they will eventually combine photonic crystals with cells. And so here's a picture of a completed device. The gray is the optical fiber, and yellow, false color, is the large semiconductor template, which tapers down into the nanobeam cavity, which is shown right here. This is a simulation of the optical field profile, and what we use is thin 200 nanometers gallium arsenide with embedded indium arsenide quantum dots, which emit at around 1300 nanometers, which is convenient for testing, for experimenting and knowing what the wavelength of this cavity is. Here are a few more pictures showing another device from a different angle. Here is the joint where the epoxy meets the optical fiber and the semiconductor template, showing a nice mechanical bond. And here I want you to appreciate how large these structures are. They're hundreds of microns large, which is really nice because I can actually hold this device, I can hold this fiber with my hand and take a look at the glint of the reflected light from the gallium arsenide membrane and know that I'm holding a nanophotonic structure with my hand and can now actually use it in remote locations. So let's go ahead and probe individual cells. We use PC3, which is a common human prostate cancer cell line. We plate Petri dishes at low density, and we take these probes, attach them to micropositioners, and essentially lower them until the nanobeam cavity touches the Petri dish surface, and then we can slide them through the side of the cell wall. And now here you see a few still life images, which don't do this justice, so I want you to look at this video right here. Here I'm taking this probe and I'm moving it around. I'm about to poke this single cell. It deforms a little bit, and then as I pull it out, it elastically returns to its original position, seemingly unharmed. Now, let's go ahead and see what's going on with these optical cavities. we built a custom zoom lens setup with optical illumination, and we collect light locally through the optical fiber, which is a large multimode fiber, so it has a large cross-section capture of the emitted photoluminescent light from the quantum dots that are within the nanobeam cavity. So here is the result. First look in the top left, you see the cavity first in air. We see this bright photonic mode peaking above this uncoupled background emission. Now, when we insert this into the cell, we see that we retain this high quality factor mode. The only difference is that it's redshifted by about 25 nanometers, which is not surprised because we went from an air cladding environment to a higher refractive index water or cytoplasm environment of the cell. Nonetheless, the cavity resonance still remains, and we can use it for future activities. Now, how do the cells take this treatment? Well, we can perform a short-term viability assay using fluorescent indicator dyes. In green tells us the live esterase activity in the cell, tells us whether it's healthy, and red corresponds to a leaky cell membrane or effectively a dying cell. Now, if you look on the right, this was a cell that was probed, and on the left is a cell that was not probed. And you see they look very much the same over here. We find that about 3/4 of the cells survive this treatment, so we'll call this minimally cytotoxic. Now, on the right column, you can actually see that here we've actually broken off one of these nanobeams and left it behind inside the cell. And even still, with this beam inside the cell for a long period of time, we still see very similar response to the viability assay. Now, the other thing is, with regards to leaving these beams behind in the cells, we can perform a much longer study. So that's what we see here. So first, look at this first row. In this first left picture, I show a single cell with a beam that was broken off into it. It's only partially inside the beam, inside the cell. This red box is a position indicator. After a few hours, the cell actually endocytoses or engulfs this full beam. Now, a very interesting thing happens about a day and a half later. After about 30 hours, the cell has grown and has gotten ready to divide. And in about 42 hours, the cell is actually dividing, and these two daughter cells are actually fighting over this nanobeam cavity, almost like two puppies fighting over a chew toy. Eventually, one of these cells is going to capture that beam, continue to migrate and divide again. We've actually observed these cells survive and migrate and divide for over a week. So this is actually a very interesting result, and we were surprised to see it, and says that essentially when we create these as sensors or provide other functionality into them, they can be used for a long period of time, and these mesoscopic probes can essentially be incorporated with interesting function over a long period of time. So let's go ahead and do a sensing experiment. So we do a paradigm chemistry of streptavidin binding to biotin and the specific surface chemistry associated with that. Now, we start off with this gallium arsenide surface. In order to protect it, we encapsulate it with ALD nanolaminate of alumina and zirconia, because otherwise this material will photooxidize in water. We perform a aminosynonization, a biotinylation, and finally streptavidin adsorption for the experiment. Now, simulation would predict a monotonic increase in the wavelength shift as we increase this organic film layer thickness. And that is no surprise due to simple dielectric permittivity environment. So let's go ahead and do this experiment. We now first start with an ordinary chip. It's not the actual probe, just an ordinary chip just to see what we expect. And we see that when we perform the appropriate chemistry, we see a large wavelength redshift, about 3 to 4 nanometers, upon streptavidin adsorption. When we omit the linker molecule of biotin, we get a much smaller non-specific binding redshift of about 0.5 nanometer. So now we repeat this process for our handheld probes. We dunk these probes inside individual solutions and monitor the wavelength after each successive step. And once again, we see the very similar results, showing that we get a large wavelength redshift upon streptavidin adsorption and a much smaller non-specific binding, showing that we can indeed detect proteins at the tip of a handheld fiber, which can eventually be incorporated inside a cell for a longer-term or dynamic cell study. So this is where we're at in this technique. We've independently demonstrated a new type of probe. We've demonstrated how it can be used and incorporated inside cells, and we've demonstrated preliminary in vitro protein sensing as a route towards real-time sensing within a cell. And so so now we're essentially adapting the chemistry, a more sophisticated antibody chemistry to monitor specific biomarkers such as PSA within PC3 cells. And hopefully this can shed a light on interesting dynamics of cell expression, protein expression, which could have informative implications for drug screening, cancer research, and cell biology. And I would like to close essentially by saying that these photonic crystal cavities are very versatile devices. They've been used in an orthogonal field for many other applications. And it's safe to assume that we can maybe find interesting uses for these inside biological cells as well. Things ranging from optical trapping within a cell, nonlinear optics, or perhaps even coherent light emission from a laser that is completely initiated from within a cell. And with that, I'd like to thank you for your attention and acknowledge the remaining crew back at Stanford, as well as my advisor, Jelena Vukovic. Thank you very much.
Thank you very much.